Well, good morning, folks. I was setting up the breakout rooms, and I did just let it randomly assign you because that seems to take less time than when I've been manually assigning people. I imagine, since we only have about half the class, that I'll be adding people to the groups. They're all somewhere between 2 and 3 people right now, but my guess is people will come in. I can see people coming in already. So I'll just be adding to your groups as you go. You may find people jumping in. So today we're going to be continuing our way through the alimentary canal and going to where lots of stuff is done, which is the duodenum, and revisiting some of the endocrine controls of both how it slows the stomach, but also how it can encourage the release and secretion of bile by the liver and gallbladder, and the regulation of the release of enzymes and bicarbonate from the pancreas. So a lot of this information I went through in the labs, but it's always good to hear it again. So you guys are going to go back over some of that. And then as we get to looking at more details of how macromolecules are broken down and what can be done with them, we're going to be looking a bit at the liver. So I have this morning, as if you saw announcements, I've already uploaded all of the previous semester's comments into a folder in the final exam folder in files. So you can find those. I also put some information up about the last midterm and the final exam. You can read that there. And then ask me in lab if you have questions rather than hear about anything about the exams. I know time is short. We only have 2 more lectures, so that's why I'd like to get started. I'm hoping we did manage to do it, that we'll get through the 15 questions today in the 3 groups, and that will leave us with 15 questions for Thursday. And then I am planning a review session, not on Thursday, but a review during our lecture time on Tuesday of next week, so 2 days before the exam. And I'm open to doing the midterm stuff and or the comprehensive stuff at that point. So let's get started. Unless anybody has a question, I'm going to just send you, it looks like people have come in, but I'm not sure why they're not showing up unassigned to the rooms. So that may happen after I open the rooms if you don't end up in a room, because I see we've had quite a few more people come into class. Okay, no questions? Let me give a second. You can put it either in chat, or I can get chat open, or just say something if you have a question. Okay, so let's go into the rooms. I've set them for 15 minutes this time, and hopefully that will still allow us to get through all 15 of the questions. And so here you go. All of us are back that came today. So we're starting with Group A, and these things are going to be useful for the practical or the lab test on Thursday. So I'm sure we'd like to hear about the other portions of the small intestine and the ileocecal valve. So who is the spokesperson for Group A?
That would be me, Professor Sana.
Okay.
So for the three parts of the small intestine, the first part is the duodenum, which is the smallest part, but it does almost all of the chemical digestion and absorption. And then the next section is the jejunum, and that is 8 feet long, has goblet cells that secrete mucus, and also absorbs nutrients and does the bile recycling. And then the last portion is the ileum, which is the longest at 12 feet long, and it also secretes mucus, and it contains Peyer's patches, which are involved in immune function. And then we also have the ileocecal valve, which is the junction between the ileum and the large intestine. And it is basically a door to block the microbes from the large intestine to prevent them from coming into.
Okay, and do you know what opens the ileocecal valve?
I'm forgetting the name of the sphincter.
Well, that is the sphincter. It is the same thing, a valve or a sphincter. I should have probably used the word sphincter instead of valve. But gastrin, which is, of course, released by the stomach early on in the process, and it's also released by the duodenum when chyme first enters it. That causes greater motility along the length of the alimentary canal. And motilin will support that, but it's released in a circadian sort of fashion, so that when the chyme arrives, it is, pushed along, as it were, by new food entering, and as it gets pushed along the length, motility increases. So sort of like the pyloric sphincter, when you get enough motility, you'll get an opening of that, brief opening of that ileocecal valve. But you did great. I think our book really stresses that the duodenum, and so you kind of want to go, oh, you got 20 feet of intestine, at least when it's relaxed in the cadaver, and what are you doing with it? Well, nutrients are continuing to be absorbed, and of those, as we go further, we'll see that fat is going to be the slowest thing to be absorbed because it has to be emulsified first. So especially as it gets down as far as the ileum, you might be still absorbing fats and bile, but the majority, as Sana said, is absorbed in the bile is reabsorbed in the jejunum, and most of the nutrients, as she said, are absorbed in the duodenum, but some vitamins are going to be reabsorbed or absorbed in later parts of the small intestine and even in the large intestine. Okay, so so what about number 2? We're going to go in looking at the duodenum in more detail. You're drawing it, and I got questions about crypts quite a bit during the drawings. What about the duodenum? Are you doing that as well, Sana, or is somebody else the spokesperson for number 2? Okay, Stephanie. Great. That's perfect. Yeah. Sometimes it's a little confusing because the simple columnar cells of the duodenum are sometimes called brush border cells, and sometimes they're called absorptive cells, but it really does kind of confuse, but it covers the whole range of what they're trying to do, because to become a monomer, which is by and large the majority of what you're absorbing, meaning you're not going to absorb glucose, you have to break it down into galactose and sucrose. You're not going to absorb sucrose, you have to break it down because it's a disaccharide into galactose and glucose before you can absorb it. So they play a role both by having a lot of transporters that we'll hear about on Thursday, and they also have a lot of brush border enzymes that Stephanie brought up. And those are the sorts of things that you're going to be studying for the lab today. We're going to do the lab, but on your own, you're going to be studying what are the enzymes, who makes them, what are they breaking down, and we're going to be using that figure in your lab book to do that. Okay, great. So the 3 levels, the plica circularis that moves things closer to the folded villi, and those folds of the villi that you can see villi in the slide, but you can't see microvilli and you can't see plica circularis in the slide. However, especially in the cadaver picture of the opened duodenum, you can see the plica circularis. And in one of the models, it's a bit harder to see because you're just looking at shadowing. Where they've made a little window in the duodenum, you can see the plica circularis there as well. Okay, so what about the absorptive cells in the intestinal crypts? Tell me especially about those crypts. Who's the spokesperson on that?
That's going to be me, Kaylin.
Okay, great.
So the absorptive cells contain enzymes in their microvilli in the brush border, and the brush border enzymes basically help complete the digestion of carbohydrates and proteins in the small intestine. And then for the intestinal crypts, they contain IELs, which are intraepithelial lymphocytes, and they're basically specialized T cells that prevent inflammatory diseases of the intestine. And then there's the Paneth cell, which are specialized secretory cells that secrete into the lumen, defensin, lysozymes, etc. And then there's the enteroendocrine cells, which are basically the sources of the enterogastrone hormones like CCK and secretin, and they're released into the blood. And then there's stem cells at the base, which are highly mitotic and constantly renewing the lining of the duodenum. And then for the layer responsible for milking the villi, that would be the muscularis mucosa, I believe, which the pulsations of the peristalsis local movements would increase contact with the chyme and it would milk the lacteals.
Great. That's perfect. So again, you can't see these in the slides, but you have to kind of know generally where they are. And again, if you're struggling with that, then we can look at those slides today in lab. But they're going to be kind of like in the stomach at the very deepest part of the villi. It sort of opens up into this crypt. So unlike the crypts that we talked about before, where you're trying to grow microorganisms, we're not doing that, although there are some highly specialized immune cells, like the PANIF and the IEL cells that she's talking about. And there's a lot of interest, especially in these specialized lymphocytes, specialized T cells in particular, that reside in your GI tract because of people who have GI tract conditions that are often autoimmune type conditions that cause gastric inflammatory diseases, those sorts of things. So that's great. And then down there are all the enteroendocrine cells we're going to hear more about. They don't have specific names like the G cells and the S cells. We're not going to worry about that. But knowing that those cells are looking at the chyme contents that make it down into the crypt and responding by releasing hormones into the blood. The defensins, lysozymes, and other antimicrobials are going to be released with the mucus towards the chyme, so that you can help control any microbes that have made it through your stomach and help protect your lining from infection. All right, great. So that's questions 1 through 3 in Group A. We're going to move a little further into the secretions released by the duodenum, and that'll be Group B. Do we have a spokesperson in that group? Is that going to be you, Jen?
I try to answer.
Okay.
For question 4, there are 5 components is secreted. One is water. The other is bicarbonate. 3 is mucus, and the next are lysosome, defensin, and IgA.
Okay, perfect. So the water and the mucus are coming from the goblet cells that you see in the slides. The immune components, defensins, IgA, lysozymes that he mentioned, those are coming largely from those PANIF cells down in the deepest part of the crypt. And what you'll notice is there are no enzymes secreted to digest things in the chyme itself. All the enzymes are on the brush border. They're actually integral membrane proteins in those microvilli. Now, into the blood, there's other things secreted. Did you also cover that, Jim?
I think just Kellen mentioned they are secreting or CCK.
Right. So CCK, secretin, GIP that we'll talk about more soon, intestinal gastric has been mentioned, motilin, which I mentioned a bit earlier about increasing motility of the GI tract, and somatostatin. Now, how did I find that list? I went to the table that's on all the hormones, enteroendocrine hormones, which I believe I gave you the number. I don't have it open to it right now, but it's something like 23.5 or something. I don't remember it right off the top of my head, but we talked about it last time. So I just went down who secretes it. There's a column, and you can make your nice list from that. All right, once things enter into the duodenum, it's going to trigger largely by the release of these hormones, secretions from the liver and pancreas that I talked about, and we'll talk a bit more about today. So what are the functions of the secretions from the liver and the pancreas? This is question 5. Is that still going to be you, Jim?
I think the liver and the gallbladder secrete bicarbonate rich the bile to emulsify fats and neutralize the chyme. Pancreas secrete bicarbonate rich the enzyme secretion, and both ductal system are controlled by the sphincter of Oddi.
Great, perfect. So, so again, we're going to revisit these hormones and these structures that I did talk about in lab. One of the things I forgot to stress on the last question was those duodenal glands that you see in the duodenum that release bicarbonate and water when the pH of the chyme enters and the pH in the duodenum is low. And those were those duodenal glands that you saw in the slides. So I thought maybe I could share the screen if this will do it. It seems like each week I have to install the app to do it. So what are the sources of bicarbonate? So that's what I was going to kind of outline. One, we just talked about the duodenal glands. Hopefully this is showing up. And those are going to be stimulated by stretch and low pH. And the next one that he just mentioned is going to be bile, which is released when CCK is released, because in order to have the bile come in, you have to contract the gallbladder and open the Oddi, sphincter of Oddi. And we talked last time, which we'll come back to, about those ductal cells of the pancreas. Sorry. Which again rely on CCK, but are stimulated by secretin. So I'm just kind of summarizing, we're going to hear more about this, but there's multiple sources of bicarbonate. In addition, the goblet cells don't secrete bicarbonate per se, but they have an alkaline mucus. So all of this, they don't actually say it's bicarbonate, but it's an alkaline mucus. So all of these things are going to kind of cooperate to bring the pH of the chyme back down towards, well, 2.7, because that's where the enzymes that we're going to talk about shortly work at. But they also, especially these goblet cells, protect the lining of your duodenum and the rest of your small intestine from this acidic, by now pH 2 chyme that's coming out of the stomach. Now, of course, they can't entirely be successful at that. So just like the stomach, and like Kaylin said, there are a lot of stem cells down in those crypts, so that you replace the lining of your stomach. I think it's every 3 to 5 days, something like that. They're all new cells along that lining. Okay, so the last one in Group B is about this portal circulation. And I'm going to try and get my tablet to turn. Didn't want to turn. There we go. So I have a little bigger space to write. And let's just remind you guys for 6 what a portal circulation is. Hopefully you know that from the kidney, but that means that there's a second capillary. And that's the capillary bed you're seeing in amongst the hepatocytes and the lobules of the liver. So that's coming in through the portal. So if we have the liver sinusoidal capillary bed. And it's coming in through that portal vein. But who feeds that portal vein? So who is going to be the spokesperson for feeding that portal vein? Is that you still, John? Nobody?
The brother from the stomach, sphinx, and intestine.
And the pancreas.
And the pancreas, yeah.
And I won't ask about the names of those vessels, but you might kind of recall them from studying the blood vessels. These were those mesenterics. Remember, we had the superior and inferior mesenterics, and then these guys were the splenic and gastric. I think there's actually has a combined name. And we didn't really talk about the pancreatic one, but the key reason I want to bring this up is we're going into the next set of questions is what's coming out of the stomach as a lot of bicarbonate, right? That's that alkaline tide. What's coming from the blood because you don't absorb things in your stomach. Well, that's not entirely true. You could be absorbing alcohols that will be directly absorbed through your stomach. And a few drugs are directly absorbed by the stomach, but generally speaking, nutrients aren't. With the spleen, what you're bringing in from the spleen are going to be the recycled red blood cells products, which we'll talk about a bit more. And the large and small intestine are going to be bringing nutrients, potentially pathogens and toxins, bile. So I'm just going to try and briefly drugs, pathogens. And then finally, the pancreas, remember, is going to be bringing in hormones. And we are going to come back to these hormones because they have endocrine cells in your pancreas. All of that's heading towards the liver through this portal circulation. Okay. So let's move on to Group C, and there's a lot of definitions in Group C. These definitions are coming mostly out of Chapter 24, the one on thermoregulation and nutrition. So who is the spokesperson for defining a few of these terms, some of which are review? So who's in Group C going to be the spokesperson?
The first question is going to be me, but we did split this one up.
What did you do? No, I didn't hear the last part.
We split up the whole group
Okay.
with the different questions. Also, the question asked for glycogenesis and glycogenolysis. Did you also want me to talk about gluconeogenesis?
Since it's not in, we will certainly touch on it, so why not? So glycogenolysis. I think these are, and glycogenesis. Okay.
I'll start with glycogenesis. Basically, it's the formation of glycogen from glucose.
Mm-hmm.
And basically, when like high ATP levels begin to turn off, the process of glycolysis, this is going to start to occur. Did you want me to get into the details about the like glucose 6-phosphate and all that? Or
No. But how about what hormone do you think would encourage this to happen?
so this is where it gets a little confusing to me, but when you have glycogenesis, the insulin, right?
Yeah.
Yeah. Because that insulin antagonizes glucagon.
That's right.
Okay.
So when the nutrients are coming in through that portal circulation, they're bringing with them, glucose is one of the things, or other monosaccharides, and those are arriving at the liver, and the liver is a great storage site for glycogen, and it provides the glycogen that you need to maintain your normal blood glucose levels. So insulin is useful to help in regulating blood glucose levels, but it also helps the liver doesn't require it to uptake the glucose or other monomers. It can do that, which most cells can't do without insulin. What insulin's doing for the liver is saying, take whatever glucose you have or other monosaccharides, convert those into glucose and form glycogen. So I want to remind you that glycogen is just a whole bunch of glucoses, hundreds of glucoses linked by dehydration synthesis into chains of a polysaccharide. So if it's another monosaccharide, if you absorb fructose, say you had some honey or you had an apple, then that has to be converted into glucose to make glycogen, and that's part of glycogenolysis as well, or glycogenesis. So notice the word genesis, that means to generate, versus the word lysis. So don't get confused with glycogenolysis. There's just the word glycogen in there. Did I spell that right? I didn't spell it right. So that would really confuse one, wouldn't it? So let me get the eraser here. I think I'm writing and talking at the same time. Got too many ENs in here. Glycogenolysis. Okay, how about that one?
So basically, glycogenolysis is when blood glucose levels drop, you're going to split up your glycogen, which is like your glucose storage, basically, to increase that. And glucagon is going to directly stimulate that.
Okay, great. But it's not the only hormone, I can't talk and write today, I guess. That will do this. So there is a table in chapter 24. You don't have to memorize it, but much of it brings back the endocrine system from 103A a million years ago. So any of the hormones that want to raise your blood glucose. So think logically about it, then that might just help you remember. If you're growing, you're going to need more blood glucose. So you better have some growth hormone. Even if you're not growing, you're just repairing cells, you're going to need to have glucose to make ATP. So growth hormone's important. If you're stressed, like you might be right now, then, all of the glucocorticoids, like cortisone, those are going to need you to raise your blood glucose. And if it's a short-term stress, you're going to be using norepinephrine, epinephrine. So even if you just get those down, glucagon is stimulated by the low levels of glucose. And those are coming from the alpha cells of your pancreas. But the others are stimulated by more global conditions of your body. What is it trying to do? And some of them are under diurnal control, like the growth hormones, so that if you might remember from 103A, you release more of it at night. So that allows you to break some of that stored glycogen that the liver is so good at storing down in order to make it make glucose, and that glucose is going to be secreted into the blood. Now, back in 103, at least in my section, and I imagine Dr. Sundar did as well, there are only a few tissues that are able to take glucose turn it from glucose 6-phosphate, which is the first step of glycolysis, back into glucose and transport it out of the cell. So one of those is the absorptive cell of the duodenum. That makes sense because anytime a glucose enters a cell, it becomes glucose 6-phosphate, the first step of glycolysis, immediately. So you don't find glucose per se inside the cytoplasm of the cell. The neat thing about that is that means it's always a downward concentration gradient to get glucose in. We are going to talk about absorption of glucose later in these questions. Once you need to get it out on the absorptive cell, then you have to take the glucose 6-phosphate and convert it back into glucose. And that takes an enzyme, which we named last semester. I'm not going to worry about it right now. It's pretty easy, glucose 6-phosphatase, but I'm not going to test you on that. The other tissues, though, that have this are the liver, because the liver has to do it in order to be the pantry of the body, in order to store all this glycogen, make it into glucose through glycogenolysis, and then secrete the glucose into the blood. And the last one that does it should be familiar as well, which is the kidney. Since it's reabsorbing glucose from the filtrate, that glucose enters the PCT cell, gets converted to glucose 6-phosphate, has to be reconverted back into glucose, and secreted back into the blood during reabsorption. So we didn't talk about that, but again, the advantage of doing that is glucose is always going down its gradient to come in, even though, as you'll see, it's a secondary active transporter that brings it in.
Professor, I have a question. Mm-hmm. So I did look at the table that you're talking about, table 24.4.
24.4, is it?
Yeah, but it says that the only ones that stimulate glycogenolysis are glucagon and epinephrine, whereas the other ones that you're talking about, growth hormone, thyroxine, cortisol, stimulate gluconeogenesis. So I'm just confused. Do I just assume that they all stimulate glycogenolysis?
You know, as far as I know, they do. I don't know why in that table. I'll go with either way in the table. Sometimes when we talked about this in 103A, it was more that I started to write the line, but it was slow. These guys are synergistic with each other. That was my memory. This would be a good thing to ask Dr. Sundar, since he's the endocrinologist here, that it's really this guy that does the gluconeogenesis and this guy that does the glycogenolysis. But because they are synergists for each of those, you know, I just included them both in there. So they didn't mention growth hormone?
They don't, like on the table, does it say that it stimulates glycogenolysis? Okay.
Well, that one I'd have to double check on, but these two, because of their synergism, I definitely would include under there, and I won't go down to that level of detail. We did that in 103A. As long as you understand you're going to be increasing glucose secretion by the liver with all four of those hormones, exact sources. I'm not
okay. That sounds good.
Okay.
Did you want me to talk about gluconeogenesis?
Yeah, why don't you do that while we're mentioning it? Oops, my tablet is being really slow. Sorry.
Yeah, so gluconeogenesis is basically the creation of glucose from non-carbohydrate sources such as fats and proteins.
Okay.
And those hormones, as you were saying, also stimulate that process.
Trying to get the words out, although hopefully, is it typing it? Gluconeogen. I don't know why it's slow sometimes. Oh, well, I'm not going to write with the tablet. I'll just stop sharing at the moment. It's just being very slow to communicate between the two things. All right, so gluconeogenesis, making new glucose. So that means you're going to take, like he said, amino acids, etc., lipids, and we will come back to that when we look in some of these pathways in chapter 24. All right, thank you, Noah. All right, so let's move on to, oh wait, we have one more question for you guys, right? two more questions. Is it going to be you still, Noah, who's doing the define the terms of lipid metabolism? Boy, I can't write anything down. It'd be really nice. So you guys are going to have to take good notes from what they're saying. Or I could use the annotator, I suppose.
It's going to be me for number 8, Dr. Kaufman.
With the whiteboard. Okay, go for it.
So, 1 second. So, lipolysis is the breakdown of triglycerides to fatty acids and glycerol. Beta oxidation is the initial phase of fatty acid oxidation. So, it converts fatty acids to acetyl-CoA, which enters the Krebs cycle to make ATP. Lipogenesis is the formation of triglycerides from carbohydrates and amino acids. And ketogenesis is the process of converting acetyl-CoA to ketone bodies, which are then released into the blood.
See if I can catch up. I'll put yours. Okay, and ketogenesis. How did you define it?
It's the process of converting acetyl-CoA to ketone bodies, which are released into the blood.
Acetyl-CoA.
All right, so so a lot of this is in, again, chapter 24, and we're kind of following along on some figures you may have seen when you looked at the lectures. And the arrows in the figures are sometimes, not all of them are not labeled, especially for lipolysis. But let's just kind of step back and look at what you've got. We're not talking about steroids here. We're only talking about triglycerides, and that's all we will be talking about at this point of digestion. So triglycerides, remember, are glycerol with 3 fatty acids, and I'm abbreviating, which you can too, fatty acids, FAs. So there are various fates, and the arrows kind of diverge in the figures, showing that once it's broken down into these fatty acids, the fatty acids then can, if you wanted to make glucose and go, this is where it'd be nice, maybe I'll try using this thing, it's going to be a little bit, this acetyl-CoA could go off and become glucose through gluconeogenesis, and it could be released, if you're the liver, into the blood to keep your blood glucose levels high. Or the liver being very metabolically active tissue, it could go through the rest of Krebs and become ATP. So this is a way you can take the energy of the fatty acid portion of the lipids that you have absorbed. As we'll soon hear when we talk about absorption, what's absorbed by fats is largely monoglycerides and fatty acids and glycerol. So we will be talking about, it's a little bit complex, but some of them are absorbed and directly into the blood, and they can go off and become, through the liver, they can become glucose and maintain blood glucose levels. And that's through that, which unfortunately, maybe I can write it quickly, through that gluconeogenesis. I didn't write it before. Now, the liver is the primary place that beta oxidation occurs in the body. You don't have that much beta oxidation by other organs of the body. So if you're going to take fatty acids and use them for either making ATP or making ATP or making glucose to maintain your blood glucose levels, the liver is the primary place that that's going to happen. Okay, let's skip over this one because the ketogenesis, and she was just doing it in the order that I did, is kind of tracing some of what happens if you make acetyl-CoA and you want to make them into something called keto bodies. Now, why you'd want to make them into keto bodies, we're going to be talking about a bit more. Some of these keto bodies can be used for a process called transamination. That is the processing of one amino acid by the liver into a different amino acid. So we will be coming back to that. Some of the other ones are particularly useful to, this isn't in your book, but to supply the brain. The brain runs on glucose, but if glucose somehow is not available, you skip and don't eat and you have low blood glucose levels, the brain can use it, keto bodies, for ATP production when there isn't a lot of glucose. So that's important. One thing that's odd is the liver doesn't use it for energy, even though it's making it, it's releasing these things, it's not using it for energy. It can use it though for, as I said, this process called transamination. Okay, and then just the middle one here, the lipogenesis, again, note the word genesis. The liver's a great fatty organ. So besides adipose, the liver stores the largest amounts of fat for just maintaining blood glucose levels. So again, being the pantry, much of it is stored as glycogen, which would be a more rapid way of making ATP than going through all these other steps that they're talking about here in order to make glucose through gluconeogenesis. The fastest way to get glucose to rise is to break down the glycogen, but that does have fat in the liver that can do that. So once again, the hormone that would increase this process of lipolysis, an increase would be glucagon. Whereas insulin would increase lipogenesis. So, and we won't worry about other hormones at this point, but when you're breaking down fat, cells growth hormone again tends to cause adipose to be broken down, and it probably also targets the liver, but again, you might want to check that table for those details. Okay, we good on this? Or anything that causes more gluconeogenesis, any hormone by the liver would increase fat breakdown as well. All right, we're into the weeds on all of these metabolic processes. So know these definitions, because I do often just ask it as a definition, but the easiest way for me, at least, to understand these definitions is know the context of where you're going to use it. For example, what hormone would be released and under what conditions. When you're not eating, then glucagon's going to increase your fat breakdown. That makes sense. If you like to diet or you want to diet, you may never like it, but if you want to, then you would be doing that. Okay, so the next one kind of goes on to the breakdown products of that I've touched on already. So let me get rid of this, and that's question 9. Who's doing 9?
Hi, Professor. I'm doing question 9.
Okay, is that?
It's Lena.
Lena, okay, great. So Lena, let me put some text up so you can type as you go. All right, go for it.
So when a person has not eaten, glycogen is needed, but if all the glycogen is used up, the body needs to tap the fat stored in the fat cells in the liver that stored triglycerides. And through lipolysis, it's going to break down into glycerol and fatty acids. The fatty acids are converted to acetyl-CoA through a process called beta oxidation, and glycerol can be converted into an intermediary through glycolysis. And when beta oxidation or catabolic pathways occur and large amounts of acetyl-CoA from glucose is generated, and if it is coming from the fatty acids direction, when the acetyl-CoA is broken down, it will tend to generate ketone bodies. Ketone bodies are primarily made from lipolysis.
All right, so we touched back again on ketone bodies that you're just talking about and beta oxidation. So the fatty acids are going to go via beta oxidation and become acetyl-CoA. And that has choices. Again, it can just go and make ATP if the liver's doing it for itself, or it can run, can be gluconeogenesis. So we'll maybe put on gluconeogenesis. You could run things backwards. You can convert that into glucose, as you mentioned. Two, you can make ATP for the liver via the Krebs cycle, or three, they can become ketone bodies. Okay, so we're good on fats. We're really, as I said, into the weeds on that. One other thing here, which, you know, we're not going to go into chemistry anyway, but maybe I'm going to put it as 4. You can also, the liver makes cholesterol, and it uses, using lipolysis intermediaries. We won't go into any of the chemistry, but later when we go through all the functions of the liver is the primary source of making cholesterol, and all cells need cholesterol, even though it sounds like a bad thing, but all cells need cholesterol to make their membranes. So if a cell needs cholesterol, the liver can help provide that. All right, going on to group D. How are we doing on the time here? Hopefully we'll make through it. So how about we're back into regulating the carbohydrates with question 10. So what hormones affect the rates of carbohydrate and fat metabolism? Again, we won't worry about exact pathways or conditions, but sometimes it's easier to remember. And I kind of we went through this before, but who is the spokesperson? We have group D, right?
Yeah, that's me. My connection is a little bit not working very well, so I might cut in and out. But for anabolism, we have insulin, which lowers glucose by increasing glucose transport in muscles, adipose tissue, and stimulates synthesis of glycogen, fat, and protein. And then we have epinephrine, which antagonizes insulin actions.
And what was the second part you said while I was typing?
Oh, sorry, epinephrine?
Epinephrine. All right. So we have catabolic pathways, meaning what you mean is gluconeogenesis and all that, right?
Yeah. And for the catabolic pathways, we have glucagon stimulates gluconeogenesis. So epinephrine is also catabolic?
Okay.
Is it? I thought it was anabolic.
Glucagon. What would you asking, Eliana?
About epinephrine. Is it part of the catabolic pathway?
I'm just sticking those together. They do actually have different targets, but since we've always just stuck the two together as if they're the same, okay, then I'm not going to ask that distinction since we've lumped norepinephrine. It's true that they kind of target things a little differently there. You're right, but we'll just stick them together.
Okay, yeah.
All right. So, great. So we've got all of these hormones that are going to encourage you to break down glycogen or turn fat and even proteins, if you need to, into glucose and increase your blood glucose levels. All right. How about generally, how does gluconeogenesis work? We've already touched on the hormones, but what's the idea of it?
So I'll be answering that one.
Okay. All right.
I said that it happens through catabolic pathways of glycolysis and the Krebs cycle in reverse. So specifically when adipose and liver cells produce glycerol through lipolysis, the liver converts glycerol into glucose, and that gets released into blood. And also acetyl-CoA, which is a product of beta oxidation of fatty acids when it's produced through the reverse steps of glycolysis. Fatty acids, they never mind. Actually, the hormones I said were glucagon, cortisone, and epinephrine. I kind of didn't answer it all the way.
Okay, great. So the general big picture is when you're doing gluconeogenesis, you're sticking intermediaries in. If it's, for example, amino acids can be stuck into the various intermediates of the Krebs cycle, but then you're running that backwards, making acetyl-CoA out of those, and then the acetyl-CoA is converted back into the end products for glycolysis. And glycolysis is run backwards until you generate glucose. So maybe I won't give examples typed out. Perfect. That's great. Thank you, Augustine. Okay, so this is a shift. Wait a minute. Where are we? Define the terms and proteins. So again, there's a bunch of terminology. We're going into proteins now. What happens to them? And so the first term I have is oxidative deamination. What happens in that? Who's the spokesperson doing the oxidative deamination?
It's still me.
Still you, Augustine?
Yeah. So I said that oxidative deamination involves an amino acid being converted into the corresponding keto acid by removing a amine functional group as ammonia, and the amine functional group is then replaced by a ketone group, and the ammonia eventually goes into the urea cycle. And transamination takes a number of amino acids that transfer their amine group to the A-ketoglutarate acid that goes from that into glutamic acid, and which will make the original amino acid become a keto acid.
Okay, so you might have, especially if you had any nutrition, understand that there are some amino acids we consider essential amino acids, and those are because you can't make them by transamination. But then there are some that you consider in your diet non-essential amino acids, and that's because you can do that. So it converts one amino acid into, I'm running out of room, aren't I? See if I can make my screen bigger so I can see it. Amino acid, whoops. Guess I'll have to do that. Amino acid into another amino acid. And the other one would be a non-essential amino acid because you can manufacture it. Okay, great. So you don't need to know examples of these non-essential amino acids, but the idea of what's a non-essential versus an essential amino acid. We'll save learning who's non-essential when you actually take nutrition, if you do. Now, when he mentioned that you have to do, in order to do this, you got to remove ammonia groups. or NH3 groups, the amino groups. And those are made into ammonia, NH4, and during that. But ammonia is really toxic substance, very reactive. So that is immediately converted into urea by the liver. And that urea is secreted. And of course, then, and I probably should have added that, ends up in your urine. So the kidney is secreting these nitrogenous wastes that the liver and other body cells, but here you have the liver doing a main one in order to do gluconeogenesis, form this into glucose, and break down that protein. Okay. So let's move into the last group. We have probably enough time because you'll notice one of the things is that all of this is focused on what can the liver do. And so I think it's all relative to SA5, the functions that the liver can do. And I don't know why this is not erasing. There we go. Okay. So who is the spokesperson for some of the things the liver stores? Which is question 13.
That's me, Manreet.
Okay, Manreet.
Okay, so our group decided that it was vitamins, minerals, carbohydrates, and proteins, as well as bile and also glycogen.
Those lipids, glycogen, and what was the last one? Vitamin, minerals, lipids, glycogen, and did you say something?
Bile.
Bile. Well, it makes bile, but I don't know that it stores it. So we won't put that under stores. So what vitamins?
I have A and D, as well as B12.
B12. So fat-soluble vitamins are especially, since it's a fatty organ, I'll put especially fat-soluble ones. It's able to put these fat-soluble vitamins into the lipids that it's storing. So the bad thing is if you eat liver, it's kind of a fatty organ, but the good thing is it's full of vitamins. The fat that you eat is full of vitamins that are being stored. And how about minerals? The only one we really talked about was iron. You mentioned that. Did you mention any others?
No, I didn't.
No, and I wouldn't worry about it. The iron is good enough for now. And then, of course, it's storing lipid and glycogen for energy or maintaining blood glucose. Maintaining blood glucose levels. Okay, great. So did we come up with 5? Well, not really there, right? 1, 2, 3, 4, 5, 6. I guess we did. But there are a few other things that get stored here. Toxins, which wouldn't be great. Drugs. So these, again, especially fat-soluble ones. So the negative thing of eating liver is, besides storing all these nice vitamins, you might want to have eat in your diet, increase if you eat an animal's liver. You may also be getting any kind of fat-soluble toxins or drugs that are being stored in the liver, because the liver plays a big role of removing from that portal blood. It removes toxins and drugs before they can sometimes get to the rest of your body. In fact, some drugs are even engineered so that when the liver takes them up, the liver metabolizes them from an inactive form into an active form. So that's another reason why some drugs are hard on your liver, like the NSAIDs, or some of them can be hard on your liver because the liver is seeing such a concentrated form of that drug. Okay, let's see if I'll probably have to erase this again. For the next question, again, focused on the liver, 10 substances it makes. Is that somebody else, or is that still you, Manreet? Maybe I'll number them. It makes or releases. Now, you mentioned bile. We already had that one. Who's the spokesperson for doing this part of you guys' questions? 14.
I was going to continue that. So I actually referred to the table you provided us in Canvas.
Great. Okay.
So there's synthesis of glycogen, so you did bile. Okay, so you did bile. So forms bile from bilirubin, synthesis of albumin, synthesis of fibrinogen and other blood clotting proteins, synthesis of alpha and beta globulins, synthesis of immune molecules such as complement and kinins, synthesis of prostaglandins, synthesis and transport of cholesterol, and recycle red blood cells, and synthesis of erythropoietin.
All right, let me see if I can catch up. Immune, complement,
Prostaglandins, cholesterol, erythropoietin. I don't.
Plasminogen, I have, well, maybe sort of unclotting, right? How about angiotensinogen? And can't spell. We could go on. And as she mentioned, there is a table. I'm going to stop sharing this in a second. I'll give you a minute if you're trying to take a picture or screen capture that. And the table she was talking about, I left open here. Let me open it. It's kind of a concept mappy thing. Sorry. And it looks like this. So there is actually a table in your book that didn't exist when I made this handout. It had it in a different way. It was kind of paragraphs, but now it's a table, and the table and this look quite similar to each other, except this is more in a concept map format, and the other one is more like, what does it do for carbohydrates? What does it do for lipids? What does it do for proteins? So these will be really useful. I would know what's on this. The other reason it's useful is like when you go back here, you can review, think about what are some of these things doing? What is fibrinogen? What is plasminogen? We covered these various reactions. What's transferrin used for? What's ferritin used for? What's hemosiderin used for? So it'd be good for review is the point I'm trying to get at. Okay, so great. That's a big, long list. Now, the next one is a bit vague. I grant you the next lecture questions. Let's stop sharing that. What is the other things that it can do? I guess it's still there. Maybe I'll just put it over here under the other category. Did you come up with anything from that same table? Someone else, not you, Jeanette.
No, it's me.
Okay, probably.
I actually just used the slides. So what I said is the liver also functions to store, I said stores vitamin and minerals again. And then I said stores and transfers iron, and it recycles bilirubin for the red blood cell cycle. And then also the excretion and elimination of hormones and drugs.
And the secretion and I'm going to put secretion. It's not really, secretion isn't a good word, but the steroid hormones are processed by the liver. I suppose prostaglandins could be considered a hormone that they're secreting, but we didn't really talk about those. But the steroid hormones, I think, is what you're referring to, where the liver can break those down. So eventually you want to get rid of a hormone, a steroid hormone that you've released. The liver's good at getting rid of those. And the last one I had was ammonia. We did talk about this to urea. So yeah, All right, and ammonia and cholesterol to bile I have. So when we talked later about moving cholesterol to your liver on the HDLs, and it'll convert that into bile. So you can get rid of some of the cholesterol as well as making some of the cholesterol. Okay, so this finishes up the essay 5 for you guys. And when we come back, we're going to go into the gallbladder, and we're going to also revisit a lot of these hormones. So I'm going to post the last set of groups today while you guys are working in lab, and it'll be 5 groups. If you guys come prepared for the answering those lecture questions, then I think you'll be well on your way to not only reviewing for the exam, getting ready for the final exam, but some of this information will be good for the lab test on Thursday. Now remember, the lab test will happen during lecture time. So that means that this first lab section and the second one, if we need to, sometimes I do, sometimes I don't, will be the lecture, the groups. So come prepared as much as you can for those, because you'll be doing the lab test first. So for the lab test, it's going to be just like the last one. There'll be PowerPoints. I think it's be great if you can both be logged in. Well, you have to log into to the Confer Zoom in order to see the PowerPoints. And if you use the chat function to ask questions, that would be good. They'll be timed. At the top of each slide, it'll say how much. Most of them are 2 minutes, but occasionally you'll have 3 or even 5 minutes for some of the longer questions. And it's pretty broad topics, as you'll see. I mean, as far as it goes all the way from reproductive structures, don't forget about those. The urinary structures, the digestive structures, the slides for the urinary and digestive systems are quite extensive. And then the last thing is what we're doing today in lab, which is some of the chemistry and how these, what enzymes are released, what enzymes are on brush border, and what do they do. So if you can study that flow chart that's in the lab exercise that we're doing today, we'll talk about it in lab more, then that would be useful for this last part of the lab. So if there are any questions, let me stop sharing and see if there's any questions in chat. So if you can show up for the lab today, having hopefully done the videos so that you have your colors and your interpretation of what the colors mean for these various biochemical tests of enzyme reactions, then I could do some background on it. We can talk about that flow chart that's in your lab manual and how to study for it. And then when we're finished, if you have any questions on the slides, the models, you can bring those and we can use that as sort of a review and going back over the things you need for your lab test on Thursday. So one of the things I noticed, and I want to run over if I give you a break, on giving feedback was there's a lot of mistakes in where the crypts are. I mentioned that already in the duodenum. There are a lot of mistakes on where is the submucosa. Sometimes people thought the submucosa was still in the epithelial area. So if you haven't looked at the comments I made on your slide drawings, then I would look now. I have not done, I don't think I've done the liver ones yet, but I will certainly do those today. So again, it's good to look at the comments on the slide drawings, because sometimes you'll have made a mistake and then you go into the test and you continue to make that same mistake and lose some points. Okay, there's one question in chat. Are the lecture sets due this Thursday? Oh, is that what it's saying on there? Well, you should have them done, but if not, then I could move the due date up to maybe the review, if that helps. I don't want to make it due the day of the test, only because I want to make sure you've got a big break now between the finishing these lecture questions and the test. So why don't I move it to Tuesday for the day of the review, and I'll make it 5 o'clock Tuesday. So what is due Thursday, though, will be what is due today is that activity for the digestive tract. And yeah, I guess that'll be good. It might be even good. Maybe I'll think about having the lecture essay that goes with the last midterm due on Tuesday as well, rather than the day of the test. That way you can really just focus on the test for 2 days. So I'll think about that. Does that seem like a good idea to you guys? You can put it in chat if you want, or just give me a thumbs up or a reaction. No, please don't do that. So what I'm thinking is, if you get all your essays done, all your lecture set questions done by next Tuesday before the review, have them all turned in, then you'll have Wednesday to study, if you want, entirely for the comprehensive part. That would free you up if you put everything, if you're a procrastinator and you put everything off till Thursday, that's not, a week from tomorrow, from this Thursday, the day of the test, then you're still scrambling to get those assignments done and not just being able to sit back and just study it all. Okay, well, I may do that then. Okay, thanks, Selena, for bringing that up. Okay, so somehow we went, and we have quite a few people who were not here at the beginning of the class. So if you weren't and you've missed lecture questions, you're going to need to get them from your buddies. Okay, I will see you in about 20 minutes then, or 15 minutes for the lab, and we're going to go over the enzyme part, and we're going to look at the lab manual and how to study about what organs are releasing enzymes or have enzymes, and then what are their substrates, what are their products, and hopefully my tablet will be a little better functioning so we can do that together. Okay, I'll see you in about 20 minutes. 15 minutes, whatever it is. See you.